Okay, so this is novel medical advancements by youth and pre-med. So this is our table of contents. The first one we're going to be reviewing is vaccines. What are vaccines? How do vaccines work? And different kinds of vaccines. And the second one is stem cells. What are stem cells? Application of stem cells and limitations of stem cells. The next one is organ transplants plus organoids. What are they and how do they work? And then the last one is credits and sources. Okay, so what are vaccines? Vaccines are a simple and effective way of protecting the body from diseases. They're usually done by injections, but some vaccines can be sprayed. Although vaccines are mostly injected into the body, the sprays are typically sprayed into the nose or mouth. Vaccines greatly reduce the risk of diseases. They work with the body's natural defenses to build protection, allowing for the body to be resistant to illnesses. Okay, how do vaccines work? Vaccines train the immune system to create antibodies. They contain killed or weakened forms of germs like viruses or bacteria. Because they only contain weakened forms of bacteria, they actually don't put the person getting the vaccination at risk of getting the disease. After the vaccine is injected, the immune system recognizes the invading germ. They then produce antibodies, which are proteins that are produced by the immune system that are used to fight diseases. Because of this process, the immune system then remembers the disease as well as how to fight it, which is how vaccines prevent you from getting sick. Because the immune system remembers, we usually stay protected by the vaccine after one or more doses. The first example of a vaccine is the COVID vaccine. There are two types of COVID vaccines in the United States, and they are the mRNA vaccine and the protein subunit vaccine. They both work differently. Firstly, the mRNA vaccine uses mRNA created in a lab. It teaches our cells to create a protein. Then it triggers an immune response, aka making antibodies, which helps us from getting sick with the virus in the future. The second type of COVID vaccine is the protein subunit vaccine, or the Novavax vaccine. This type of vaccine contains pieces of COVID-19 and teaches the immune system to respond to the virus. Um, then the immune system remembers and knows how to fight it in the future. And the last example is the HPV vaccine. This vaccine helps protect the body against a virus called human papillomavirus, aka HPV. This is spread through sexual activity and can cause cancer. This vaccine protects against genital warts and most cases of cervical cancer. It protects against the mouth, throat, head, and neck cancer caused by HPV. The suggested age for getting the HPV vaccine is 11 to 12. This age is quite young because once a person has the virus already, this vaccine is actually not guaranteed to help the body clear the virus. The side effects tend to be mild, with the most serious effects being dizziness and fainting. However, staying seated for 15 minutes after the virus helps the side effect. Now moving on to stem cells.
Okay, the second advancement that we would like to cover today is the application of stem cells in the field of therapeutic medicine. Stem cells offer endless possibilities in the field of medicine because these cells have the ability to become a wide range of specialized cells. Stem cells, stem cells are what we call non-differentiated cells, which simply put means that they can become any cell that they want through the process of cell division. This diagram on the right showcases how a single stem cell can become into muscle cells, cardiac cells, blood cells, neurons, and the list just continues. Furthermore, stem cells also have the ability of self-renewal, which basically just means that they can divide again and again to produce more and more copies of themselves, which is useful for growth or tissue replacement when cells are lost or damaged. As such, stem cells have taken the spotlight and become one of the most studied and active areas in research today. First, let's talk about where and how these cells can be obtained before we learn about their wide range of functions and applications. These fascinating stem cells can be broken down into two categories. There's adult stem cells, which are present throughout the human body. They're typically found in microenvironments called stem cell niches, which are lounging areas for stem cells where they await their turn to differentiate into other different kinds of cells. These stem cell niches can be thought of as, um, the stem cells in these niches can be thought of as unemployed cells that are waiting to be summoned by the body to take on a role which they stick with for the rest of their lives. Stem cell niches are very prominent in the bone marrow, and through that, our body is able to generate more red blood cells when the current ones die. Adult stem cells can also be found in the brain, the skeletal muscles, the skin, and the liver. However, these cells can be difficult to obtain because they remain inactive for most of their lives until our body summons them to repair or grow new tissue. And also, while these stem cells have a decent ability to become into any cells, they do still have some limitations because they cannot just become any cell that they want. This leads us to the second type of stem cells, which are embryonic stem cells. Like their name suggests, they're derived from an embryo, which typically occurs a few days after fertilization. These cells are typically totipotent or pluripotent, which basically just means that they can become basically any cell that they want in our human body. And unlike adult stem cells, they which have fewer opportunities for therapeutic purposes because of their inaccessibility and limitations. Embryonic stem cells are very useful. They could be used to produce regenerated tissues, such as skin for people who've suffered burns, and they could also be used as a means of healing diseases such as type 1 diabetes, where a particular cell type has been lost or is malfunctioning. They might also be used in the future to grow whole organs and replace tissues such as the heart tissues and the kidney tissues. Um, one of the most common um, One of the most common applications of stem cells is to cure Stargardt's disease, which is also known as Stargardt's macular dystrophy, which is a genetic disease that develops in young children due to a recessive gene. The result of this disorder is malfunctioning photoreceptive cells in the retina, which basically impair an individual's ability to see, and it causes someone's vision to progressively become worse, and to the point where they can be registered as being blind. So basically, researchers have developed methods for making embryonic stem cells develop into retina cells, which was initially done with mouse stem cells, which were then injected into the eyes of the mice that had a condition similar to Stargardt's disease. And since these cells were not rejected, and nor did they develop into tumors, researchers were granted permission to test these cells on humans, and they were surprised to see that the treatment actually worked, and the stem cells attached to the retina and remained there for the rest of their lives. And this gradually improved the patient's vision without inducing any harmful side effects. Another application of stem cells is to treat leukemia, and this is not limited to just embryonic stem cells because adult stem cells can also be used to cure this. Basically, adult stem cells from donors have the possibility of being used to treat this disease, and some of you may not know what leukemia is, so we'll cover that first. It's a well-known type of cancer that occurs due to uncontrolled cell division, and if it affects over 250,000 people annually. And it involves the production of abnormally large number of white blood cells, which can cause to the development of tumors, and it could also lead to the deterioration of someone's health. To cure leukemia, the cancer cells in the bone marrow that produce large number of white blood cells must be destroyed. This can be done through chemotherapy, which kills the cell, However, but to also remain healthy while the treatment is ongoing, patients must be able to produce white blood cells that are needed to fight the disease. But since the stem cells that produce the white blood cells are killed along with the cancer cells, the following procedure is used. So first, a large needle is inserted into a bone where stem cells are extracted from the fluid that's within the bones, and they're stored by freezing them. And then these adult stem cells have the potential to produce some different types of cells. In the case of leukemia, they're focused on generating red and white blood cells. Once a high dose of chemotherapy is given to the patient, the cancer cells are killed off in the bone marrow, and then the stem cells are returned back to the patient's body, which allows them to reestablish themselves in the bone marrow to produce red and white blood cells, which not only kills off the cancer cells, but it also maintains the red and white blood cells, which can then help them heal from the disease. Stem cells have obviously captured the imagination of various scientists for obvious reasons, and their study offers new avenues for treating conditions such as heart disease, diabetes, neurological disorders, and spinal cord injuries. These therapies hold the potential to replace or repair damaged tissues, restore function, and improve quality of life for patients. It is simply remarkable how these small cells have so many capabilities. However, we must also address the ethical concerns that are present with the use of these stem cells because the research into stem cells, specifically embryonic stem cells, is considered unethical and very controversial for a variety of reasons. Since the extraction of stem cells from an embryo leads to the destruction of potential life, which I've quoted here because there's no general consensus on whether an embryo is considered to be living or not, research with embryonic stem cells continues to be a topic of debate. However, the bottom line is that the unethicality with embryonic stem cells does not lead to a dead end in stem cell research. Scientists have managed to develop these cells, known as induced pluripotent stem cells, or iPSCs, which are developed in laboratories, and they function with a similar potential to embryonic stem cells, and they reopen the gates to stem cell treatments because these cells do not present the same ethicality concerns that are present with embryonic stem cells. The field of stem cells has an ever-growing potential, and if you're interested in these ideas, biomedical engineering or research may be a great career consideration for you. Now we'll be moving on to organ transplantation and organoids.
Okay, so first we'll be looking at what is organ transplantation. Organ transplantation is a process by which organs from eligible donors are collected, transported, and transplanted into the bodies of recipients. Individuals can elect to be organ donors in the case that they become brain dead or otherwise pass away when their organs can still be collected. Some organs, like kidneys, can come from living donors. Patients who need organs due to illness or injury are put on a list of waiting recipients. When an organ becomes available, it will go to a person on the list determined by blood type, genetics, and other factors such as medical urgency. But a person can be on the organ transplant list for many years and may never receive an organ. Next slide, please. Okay, now we'll be covering how does organ transplantation work. Organs are respectfully recovered from the bodies of donors by a specialized surgical team. Then they are delivered in special transport containers to the recipients. Organ transplant surgery occurs as soon as possible after collecting the organ or tissue so that it is still viable. The procedures are complex and often take hours to perform. Next slide, please. Okay, finally, we will be talking about how successful organ transplantation is. Depending on the type of organ, its complexity, and its vitalness to human function, the success of organ transplants ranges from about 88 to 97% worldwide in terms of survival 1 year after the operation. Modern medical advancements, such as organ perfusion systems, which pump blood through organs and make them viable for longer, have made organ transplant much safer and more effective and greatly reduced the risk of complications or rejection. Next slide, please. The final topic we'll be covering is organoids. And first, we'll be looking at what are organoids. Organoids are lab-grown tissue clusters derived from stem cells, which we talked about earlier. These stem cells can be collected from the skin and are placed under specific lab conditions that allow them to self-organize into miniature organ models under 1 square centimeter in size. Organoids have a multitude of potential uses in the medical and science research fields. Next slide, please. How are they created? Organoids can be created in several different ways. Generally, samples of tissue are taken from patients and used to derive undifferentiated stem cells, which can grow into any type of tissue in the human body. These cells are exposed to specific conditions to create a certain kind of tissue. For example, an organoid made up of neural tissue. These conditions include specific sugars, vitamins, and minerals. The sample is then able to develop independently with appropriate laboratory conditions, essentially growing itself. Next slide. Okay, finally, we'll be covering potential uses for organoids. Organoids have many diverse uses in the fields of science and medicine. Even though they are not part of an organism, the cells of organoids are the same as specific tissue types in the human body. Because of this, they can be used to study the ways that diseases affect the brain and to affect the body and to explore the potential side effects of new medications. Brain organoids can be studied to observe less understood parts of the brain, which is difficult to observe in action. Brain organoids can also be used to study human development and the impact of developmental conditions. Next slide. Okay, thank you for listening, guys.
Yeah, that was a wonderful presentation. I know this group got started really early to make sure that they could like explore all the different novel medical advancements that they were interested in presenting and kind of gathering like thorough information to present to you guys. So thank you for the good effort. If you guys have any follow-up questions, feel free to put them in the chat. But I feel like this group really highlighted a lot of important medical advancements. For the first one, like vaccines are obviously very important. It was very prevalent when during COVID, when everybody was getting the vaccine so that we could be safer and kind of transition back into normal life post-COVID. And the use of RNA has been a very popular advancement to study in the medical field. So people have been looking at RNA for vaccines, obviously, but also microRNAs to study like diagnostic tools by looking in the blood for like more non-invasive diagnostic tools. And there are many studies that are looking at RNAs and microRNAs in any field of research and medicine. And then for the lymphoma part, I think now there are new therapies also, like not only chemotherapy, but also T-cell, CAR T-cell therapy, which can treat the cancer patients without leading to hair loss. And they're more gentle compared to chemotherapy because chemotherapy only kills like the cells that are highly proliferative. And so kind of looking for genes that are upregulated or downregulated in different types of cancer has also been like a really popular area research. And then and the organ transplants, I think some people have tried like looking at organs from different animals because it is very difficult to get like an organ donation since there are so many people on the waiting list. And I think the advancements in organ transplants and organoids are definitely very important. And something interesting that I've learned recently is that to kind of diagnose and take biopsies for diagnosing like lymphomas, people actually like the doctor during your visit can actually just like poke a needle into your spine and collect the cerebrospinal fluid or CSF from the brain and spine and kind of take that sample to the lab and biopsy that. And they can also poke like a needle into your bone marrow and extract blood and kind of see if there are cancer cells in there that way. So there are a lot of like newer procedures that are less invasive and can be done like just in a normal outpatient setting without having to have the patient like stay in the hospital. So I definitely think that this presentation covered a wide range of novel medical advancements, and in the coming weeks we'll also be talking about CRISPR, which one group was very interested in presenting. And that was actually discovered very close to where a lot of our interns are from in UC Berkeley by Jennifer Doudna, and she won like Nobel Prize for that. And I think that's like a very promising area of research as well for a lot of different types of like diseases. And it's also important to consider ethics as was brought up in this presentation. So if anyone has any like questions or comments, feel free to put them in the chat. But yeah, it was a very strong effort from this group, and I think this was a very good presentation. And then if no one has any questions, you guys can feel free to go and have a good weekend. And I hope you all enjoyed like spring break for those of you who are on it. And also, I think earlier this week there was like an eclipse, so I hope that was well for everyone.